Hello and welcome back, friends. In this particular video we'll be talking about uh, the post-transcriptional modification of mRNA, and among those post-transcriptional modifications, one of the major modifications that have taken place into the mRNA is called the 5' capping and the 3' polyadenylation. So what happens, what is the big picture, is that we are having DNA inside the nucleus. Now that DNA is transcribed into mRNA utilizing the code that is present in the DNA, which is transferred to the mRNA. Now the mRNA is becoming the code holder. Now after making the mRNA, in case of eukaryotic system, what is happening is that mRNA must leave the nucleus and come to the cytoplasm, because cytoplasm can provide the workbench for the protein synthesis using that code that is present in the mRNA, and those workbench are ribosomes. So that mRNA must come to the cytoplasm. Now before coming to the cytoplasm, that mRNA, which is first produced from the DNA inside the nucleus, must be edited or modified in some way. In previous discussion when we talked about splicing, I've told all these things. Now I'm just recapitulate all these things, that in eukaryotic system the first mRNA that is being made from DNA is not the actual code holding sequence to be translated. So we need to modify this to start the translation process, to start the protein synthesis. So for this purpose, whenever it comes down to the site, before coming to the cytoplasm, it must be edited. Now during its movement, during its production, as the production is going on, as the mRNA is transcribed from the DNA, it is being modified. The modifications like 5' capping, 3' polyadenylation, and also after that, when the whole sequence is being transcribed or the transcription is going on in speed, then also splicing starts to occur. In splicing also, introns are cut out, exons are ligated with each other to make the functional copy of the mRNA. We call it the mature kind of mRNA. But the first type of mRNA that is being produced, the crude mRNA that is being produced, is not what it can what can produce proteins. Okay, so that's why we need to do these modifications. Now the modifications like 5' capping and 3' polyadenylation having a particular significance to be happen, because this kind of modifications will in turn stabilizes the mRNA. Now why the mRNAs need to be stabilized? Because the mRNA is produced in the nucleus, it needs to go a journey from nucleus through the nuclear pore into the cytoplasm. Now during this time it needs to be very much stable. Now the stability is provided by this 5' capping and also 3' polyadenylation. Though it is a region of active research, but still this is actually seen that this process is giving them the stability. Now in this particular video we'll be talking about what is 5' capping, what is its significance and what is its importance, and the mechanism of 5' capping. Okay, so here in this picture I am having the 5' capping structure. Now before that I must tell you what is in a sense 5' capping. So say we are having the DNA. So let us consider this is our DNA. Okay, now from this DNA we have made the mRNA. Now the mRNA what we've made, it's a crude mRNA, remember. So the mRNA is produced. So say mRNA is transcribing from here. So this is the mRNA, it is transcribing. So we must put our enzyme polymerase, RNA polymerase sitting on here. mRNA is being produced. Now this mRNA which is being produced is in the direction from 5' to 3', because the transcription is having a direction 5' to 3'. So it is produced from 5' to 3'. So as it is producing, we are having 5' of the mRNA free. Now this 5', whenever it's getting free, 5', now this tells this mRNA now it's time for the modification of the 5'. So this is a kind of first kind of chemical modifications that are being done when the mRNA is being produced. Now modification will be done in this 5' end. Now after the completion of this total synthesis of mRNA, 3' end also become free, then the 3' end will be modified. Okay, we'll be seeing it later videos, but in this video we'll be seeing the modification at the 5'. Now in this case at this 5', it is modified utilizing a special base. not a special base but a regular base adding in a special manner. Now we know that in case of DNA and RNA we are having four, actually overall 5 different kinds of bases we can see: adenine, guanine, thymine, cytosine and uracil. Now in case of RNAs we are finding adenine, guanine, cytosine and uracil. Now in case of this 5' modification, the base which is most important is G or guanine. Now this G base is added to the 5' terminal or 5' end of the developing mRNA to protect the mRNA from damage, to protect the mRNA from being cleaved out by exonucleases, right? Because exonuclease can cleave nucleotide sequences from the terminal ends, whether it is a 5' end or a 3' end. Now we can see there are different mechanisms to protect the mRNA from both the directions 5' and 3'. And to protect it from the 5' end degradation, we put this G into this 5'. Now question can come: exonuclease can cleave the terminal nucleotides, right? So whether it is a G or it is a T or it's a or it is a C, whatever, it must be cleaved. It can be cleaved by this exonucleases, because exonuclease can cleave any of these nucleotide sequences, and that is correct. So we need to be careful when we are putting this G or guanine. This attachment of guanine to the 5' end may not be the regular phosphodiester linkage, because the regular phosphodiester linkage can be easily cleaved by most of these exonucleases. So this is a very important point, and from where we will be focusing on to that is that this guanine residue which is being added to the 5' end is not added utilizing the regular phosphodiester linkage. It is utilizing a different kind of linkage. So that's why I have talked that a regular base attaching in the irregular way. Okay, so let us now focus on to the picture, this big picture what we am having here. Now in this picture we can see this is a phosphodiester backbone. Just let's begin with this phosphodiester backbone. Now here we are considering only two bases. So this is first base, this is the second base. So first base, so say here is our first base. So base number 1, here is the base number 2, base number 3, and so it is going on like that. Base number means the nucleotide, nucleotide 1, nucleotide 2, nucleotide 3, like that. Now here this is the first base, so it's from the first nucleotide. Now this from the first nucleotide, it could be anything. It could be adenine, it could be uracil, it could be anything else. Now at this point what we are having this phosphodiester linkage. Okay, just look at this phosphodiester linkage. Now if I mark each of these carbon numbers, so it is 1, it is 2, it is 3, it is 4, and this is 5. In the similar way, 1, 2, 3, 4, and this is 5. Now the linkage of phosphodiester is normally between the 5' carbon CH2 here with the 3' phosphate. So this kind of linkage we can see. This is called the phosphodiester. In both the cases, 3' phosphate of the first base will be attached to the CH2 which is present in the 5. So the 5 number carbon of the second nucleotides. That's how the linkage is done, and each of them are having importance. The carbon 1 is having the importance of holding the bases in all these cases. If we draw further, we can see carbon number 1 is holding the bases. Carbon number 3 is providing the phosphate, is binding with the phosphate, and utilizing the phosphate is also binding with the next base. We can see it here. Okay. Carbon number 5 is also involved in the binding with the other bases via the phosphodiester linkage. So carbon number 3 and 5 simultaneously providing the position for that base, the position for the nucleotide sequence to be attached with each other via the phosphodiester linkage. Now on the other hand, this last one, or this carbon number 2, it is responsible for the production. It is responsible for telling us whether this is a nucleotide of deoxynucleotide or a ribonucleotide. Okay, now ribonucleotide, in case of ribonucleotide there would be oxygen. In case of deoxyribonucleotide there won't be oxygen present there. So by looking at carbon number 2 we can tell what it is. This is a simple way of looking all this sugar, because the sugar is the central point, so it is providing this platform for attaching with different things. So it is very important. Now this is a phosphodiester backbone. Now The regular linkage is between 3 and 5. Now at this terminal point, remember we'll be adding this G. We'll be adding this G at this terminal point. Now this linkage will be somewhat different, right? Now in this case we can see this is a nucleotide, the terminal nucleotide, where we are adding a G, a base which is G. Okay, so it's not singly the base, we are adding a whole nucleotide, and that's quite logical because we cannot add a base just like that. We must have the whole nucleotide sequence to be added. So this is simply GTP, guanosine triphosphate. So we need guanosine at the form of GTP or guanosine triphosphate. So it comes in, so you have G with 3 phosphate groups. So that means we are having G with 3 phosphates. Now at the terminal point also, the CH2 is bound with also 3 phosphates. Remember, it is bound with 3 phosphates because 5' phosphate, because this is the first one, so we are having 3 phosphate. It is not attached with any other, so it is having 3 phosphates. And also the GTP comes with 3 phosphates. Now what happens, this GTP usually cleaves. 2 phosphates will move away, producing pyrophosphate. 1 phosphate remains. Now using this 1 phosphate and 2 phosphate from 5', it will be added to this 5' terminal. Now how it is added? Now here look at the linkage pattern. Now in previous cases of phosphodiester linkage, what we can see, the linkage is between the 3' and the 5'. 3' of the above base, 5' of the below base. Now this addition is between 5' and 5'. So let me mark this first. So this is number 1 holding the base, number 2, number 3, number 4, and this is number 5. So here comes the importance. Here we can see 5' is attached with 5'. 5' phosphate is attached with 5'. So the carbon number 5 in both the cases, the very first base and the guanine, which is to be added as a capping, both are attached with each other by the 5' phosphodiester linkage. This is also phosphodiester linkage. This is a 5'. Now in case of other phosphodiester linkages, we can see it's a two ester bonds. That's why we're calling the phosphate diester bond. Now in this case we can see there are 3 phosphates instead of 1. So this is also another importance. So the 3 phosphate groups, 2 from this and 1 from the GTP that just came in, will be attached with each other. I'm giving this, I have drawn this phosphate like that, but it's actually attached with oxygen bonding like that. So this is the important linkage between first base and GTP, which is just added. This is the first kind of attachment and the modification that we can see in case of 5' capping. So you have successfully added guanosine to the terminal point of the 5' end. Now what is to be done? Now further modifications also occur. What kind of modification? Along with this, the guanosine that is present, it is also modified. So this is not a regular guanosine also. This guanosine is methylated at the N7 position. So the nitrogen 7 of the guanosine is methylated. This is another modification. So along with both the things, this linkage between 5', 3 phosphate groups, and then N7 methylation, along with these two things, we call it 5' capping. So both the things combined together is called 5' capping. So if I am making a barrier from here, so if this is the barrier, then this part is the total new thing, and this part is providing the cap. So this is the cap portion of this mRNA. It is called the 5' capping. So now if I draw the cap here, it will look like something like this. So this is the cap. So 5' capping is done. Most of the systems we can find 5' capping like that. And remember, this thing happens only in case of eukaryotic system. Second thing, this capping is done before the mRNA leave the nucleus. And also third thing, this capping is done not after the whole synthesis of mRNA. Just after synthesizing the first two, three, four, not first few bases, capping event begins. So as the polymerase is synthesizing further mRNA, capping is already done. That's why I have made the picture like that. Okay, so these are the important features of this. Now most of the cases we can find capping like that, but also in some cases we can find further modifications in this capping. Now if we are having most of the modifications like that, we call them cap 0. Okay, so what we call them cap 0. So this part is called cap 0 or cap O, because this is the first kind of modification that is discovered and can be seen vigorously in many species. Now let's consider further modifications. Further modifications along with this, remember, because this is the characteristic capping, this must be present in any kind of capping, but along with this we can have further modifications. Modifications like this one. So the bases, the very first base that we can find, this base, N6 position of this base may be methylated sometimes in some species, in some organisms. Now if this kind of modifications happen, then along with, obviously along with this first type, this type of modification must be there. Along with that, sometimes in some species we can have this type of modification like the methylation at the N6 of the first base. Along with both the things, we call them cap 1. Okay, done. And also in further modifications we can find the portions of second carbon, both the case, is modified with the methylation. So remember, first 2 or 3 bases, second carbon of the sugar is modified, it is also methylated like that. Okay, so this part along with this part are common features, but sometimes N6 of the base 1 can be modified via methylation. We call them cap 1 structure, but remember this small modification is not cap 1. Along with this cap 0, if this modification happen, then along with everything, combining everything, we call them cap 1 structure. So now the cap structure will be this part instead of this part. Okay, sometime also modification can be seen in this base. This base can also be modified. This base can also be modified. Now if this base is modified, and again the second carbon of the sugar is modified by methylation, then along with this cap O, we can call it cap 2 structure. So you can find cap 0 is the most common, vigorously found, but along with this kind of modifications we can have modifications in N6 methylation of first base. Whole thing is called cap 1. Now if the modification is cap 0, then the N6 of the first base methylation and obviously N6 of the second base methylation, all these things are happen and can be seen, then this whole thing we can see, we can tell this whole thing is being capped. So this whole structure will be called cap 2. Now usually capping is restricted to this first 2 or 3 bases. Whatever kind of further modifications that has been taking place is happen in first 2, 3 bases, not more than that. Okay, so usually we can find cap 0 most of the case, but sometimes in some eukaryotic organisms we can find cap 1 and cap 2 structures, which are rare, but we can find this kind of structures. Now this is what is capping. Now if we see the mechanism of capping, we want to look at the mechanism of capping, what it will be. So let us look at the mechanism of capping. So I hope you understand what is capping. So let us do a next video about capping mechanism. So I hope you've enjoyed this video and learn how capping is done and what is the structure at the capping point and what are the differences between the addition of this GTP at this terminal end. I hope this helped you. Thank you.